Squamous cell cancers in the head and neck originate in the mucosal linings of the upper respiratory and digestive tract. The mucosal linings and the skin protect the body from toxic substances and from infection by microorganisms. These squamous tissues consist of cell blocks or clonal units, each produced by a single stem cell. The stem cell divides to produce transit-amplifying cells, which divide more actively. The stem cells and transit-amplifying cells are found within the deeper squamous tissue layers, above the basal membrane. The daughter cells of the transit-amplifying cells move slowly upwards to the surface, where they take on a flattened shape and are finally shed. The whole process takes a few days. The continuous assault from carcinogenic substances, such as cigarette smoke or alcohol, can damage the DNA of the cells, as can viruses such as the human papilloma virus. If the DNA of a stem cell is hit by a carcinogenic substance or a virus, the cell normally responds by repairing the damage. However, such damage can sometimes be left unrepaired, and all of the daughter cells in the stem cell's clonal unit will have exactly the same genetic change in their DNA. Now a so-called patch is formed. Turning to a top view, several clonal units can be seen, two of which have become patches of genetically altered cells. Continuous exposure to carcinogenic substances or viruses results in cumulative damage to the DNA, and eventually an important line is crossed. Increasing DNA damage overwhelms the normal control mechanisms of the stem cell, resulting in uncontrolled division. The neighboring normal clonal units are laterally displaced, and a large precancerous field arises. Despite their lateral expansion, these fields are not malignant tumors, since their cells are still unable to invade the underlying tissue. Precancerous fields can remain in this state for many years. Special techniques enable these patches of genetically damaged cells to be visualized under the microscope. The same methods can also reveal the much larger precancerous fields, which can grow to up to 10 centimeters in diameter. Further genetic assaults on the field of damaged stem cells might change one of them into a full-blown tumor cell. Its rapidly dividing daughter cells can now invade the basal membrane. The growing tumor then threatens the vital structures in the upper respiratory and digestive tract. Eventually, tumor cells enter the lymph and blood compartments and spread throughout the body. Once diagnosed, a tumor is removed by the surgeon. However, surgeons often fail to remove all of the precancerous field, which is generally larger than the tumor itself and invisible to the naked eye. Any lesions left behind can easily produce recurrent cancers. If these arise within 2 centimeters of the primary tumor, and within 3 years of treatment, they are referred to as local recurrences. Cancers not meeting these criteria are called second primary tumors. Only recently has the clinical significance of these fields been recognized.